Prof. Tagwire, how are you?
Okay. Hello. Can you hear me? I hope you can hear me. Okay. Thank you very much, Professor Jiri. So I really will not be talking about specific innovations which are in the pipeline, but what I wanted to do was to just quickly go through one product. Is a story which those who have known me for years have heard me share on a number of fora. And then what I was hoping we would do as we are going through this story is to then highlight some opportunities and perhaps some things to think about and look out for as we are moving in the heritage-based innovation in the health sector or health sciences. So I've just titled it Opportunities in Heritage-Based Innovations in Health, the case of Boophane. And Boophane is a plant that I have been working with for several years, and I wanted to give it as probably a template to then bring out some other opportunities which are there. Okay, so just quickly moving forward, just a few definitions as we start, because this story, it hinges a lot on defining and differentiating between what a poison is and what a medicine is. And even during these times when, we are spending a lot of time where we are hearing about different plants being used for COVID-19 and other things like that, the issue of what a poison is, what is harmful, just jumps in quickly. So just quickly, a poison is a substance that is harmful or lethal to a living organism, put very simply. And a medicine is a substance which specifically promotes healing when ingested or consumed in some way. And then for the purposes of this particular presentation, whenever I've got a question mark, it shows uncertainty or doubt. And as I said, this is going to be a story that I'm going to tell. And by definition, a story is a sequence of real or fictional events. This one is going to be a real one, or an account of such a sequence. So hopefully by the end of this story, which I hope to move pretty quickly through in the next 15 minutes, I want to have demonstrated, research that we have done on one particular plant. There are several others, and also to highlight some potential opportunities. And then also would like to really suggest a possible model or a blueprint that can be used for the development of medicinal plants, which can ultimately be registered as complementary medicines to start with, as well as maybe talk about some of the opportunities as we move forward. So my PhD and past research was largely in the area of clinical toxicology. So it means I have an interest in anything which is poisonous as it relates to human beings and the management of poisoning, as well as how a person who's poisoned would present to a hospital. And that was what my work was largely on. And then as I was doing my work, I came across a certain group of compounds, the traditional medicines, which appeared to be responsible for poisoning in Zimbabwe when people were using them, either for the treatment of some condition, or if somebody, made a mistake and picked up a wrong plant, and something like that. And it was during this time, when I was doing my PhD, that I first came across this plant, distica, otherwise known as mzepete, which is the plant that we're going to be using when we're talking about issues of opportunities and potential for drug development. And it was during when I was going through cases of people who had been poisoned that I came across some who had been poisoned after taking this particular plant, which is known to many people. So that plant actually is known to be a poison. The name itself, Boophane, it talks about a cattle killer. And there have been cases of people who have been poisoned from this plant in the literature, including here in Zimbabwe. But also, I noted that some people were poisoned from these plants even after taking it for medicinal purposes. And that's one thing probably that we can just learn about or think about, that there are some herbs which are there, which may be effective, but it's always a good idea for us to try and find out, what the potential consequences are of taking that herb incorrectly or taking too much of it. Is a point perhaps with the COVID crisis which is happening at the moment? We know that Zumbani is the in thing, and people are taking Zumbani as a tea. They are also taking it through the respiratory tract via Kunatira. But remember, when Zumbani was being taken as a tea ordinarily, maybe somebody would take it twice, maybe 3 times a day. But what are the effects of this Zumbani plant? Perhaps if somebody takes it 10 times a day, but Aparkunatira at the same time, they're also drinking it. Also, Zumbani, what effects does it have in pregnant women? Because at the moment, we know that everyone is taking Zumbani, whether for as a preventative measure against COVID or for treatment or management of COVID. But what are the consequences of those? So it's just one thing which probably just should come up and we should think of. So from the definition that we had of a poison, distica, which is a plant, is a poison. But then you see, in the world of toxicology, which I belong to, there's a wide definition which is held out, which anyone who claims to be a toxicologist or who has ever done toxicology, if you ask them this question, they answer you otherwise, you start doubting their credentials. The man called Paracelsus, he defined poisons as this. He said all substances are poisons. There is none which is not a poison. The right dose differentiates a poison from a remedy. And that's a very important thing, to take note. You take enough paracetamol tablets, you're in the domain of pharmacology. You take 2 paracetamol tablets for a headache away. You take 50 paracetamol tablets, then you are now moving into an area or a domain of toxicity where somebody can actually be poisoned. He can actually die in the process. Okay. So when I then came across this plant, there were a number of questions that came in, and I called them what if questions. And one thing, which the heritage-based type of research does is we've got a lot of what ifs, because over the years, people have tended to concentrate on individual molecules. They have not spent as much time looking at the actual plants. And that's a huge opportunity with regards to doing research, finding new molecules, finding new treatment modalities. So with us, because we knew Boophane was toxic, and it seems like people who died from this plant, died as a result of respiratory depression. They got into a coma and basically failed to breathe. We thought, hey, what if, using that principle that Paracelsus was mentioned, if we can calculate what a safe dose is, then perhaps ultimately we could be able to come up with medicine. And that's basically what we did. We started off by looking at the toxicological aspects of that particular plant to try and find out at what doses do people or do animals get poisoned, so that we could calculate backwards a dose which should be safe to use. And then we try to find out if it can actually work for any conditions. So we did that, a number of studies, most of which we actually were able to show out and publish. And one of the things that we then came up with, which is another opportunity, is we came up with a potential mechanism of toxicity of this plant. We observed something which is referred to as retropulsion, whereby an animal moves in reverse. And for those who have done, animal physiology or biology, you know that if an animal moves in reverse, it normally means that you've got activity of either serotonin, dopamine, that the animal is having hallucinations. So we then speculated for that particular plant that the toxicological effects that we saw were as a result of an overdrive of a certain molecule or a neurochemical known as serotonin. And then we set out to actually test that hypothesis by using a specific serotonin antagonist called cypriot. And we actually managed to reduce the mortality in the animals. So we're quite excited because now we could come up with our own mechanism of how this plant works, which brings me to another opportunity from this particular thing. Most of the times, the textbooks that we read, the narrative that we get is a narrative that the Westerners have come up with. And one thing that research in this particular area in heritage-based science will do for us is that it will also help us to come up with our own narratives. you know, when we read, we are told the mechanism of action of this particular plant or of this particular drug is A, B, C, D. But the moment that we are able to do our own heritage-based research, then we can now be able to tell the world that from our studies, we have found out that this plant actually works in this particular manner, which would is a good thing, for us. So the first step was to find out the toxicology. The second step then was to try and also understand the pharmacology, how this plant actually works. And we did numerous studies, you know, ranging from studies in anxiety, depression, neuropathic pain, memory, as well as other things. And we ultimately were able to come up with a number of potential conditions where this particular plant can be used. And that's one thing which is again exciting about, is an opportunity about the heritage-based approach to health, is that you see these plants, they contain lots of phytoconstituencies. One plant could have, I don't know, over 100 different secondary metabolites. And what it means is that then the approach is, if you've got paracetamol, I know that paracetamol could be used largely for pain, largely, and fever. But when you're dealing with plants, because they've got a plethora of other compounds, there's an opportunity to actually be able to explore a number of different conditions all at the same time, as we did, and to be able to actually demonstrate that the plant might actually be able to work for that. So we then had, you know, data which was toxicological, as well as preclinical pharmacological data, which is extremely important if you are going to register any compound with our regulator. So we proved safety in an animal. We knew the doses that would be used safely in animals, and there's a formula that you can use to calculate what doses to actually use in human beings, which we subsequently used. And we showed preclinical efficacy, not in human beings, but then in animals, we could show or demonstrate for a number of conditions that this plant can work. But then, you know, with some other questions which came in, you know, we wanted to find out, you know, what were the actual active ingredients. We knew that alkaloids were involved, so which alkaloids were there, and could we isolate any alkaloids that we could use as markers? So we then went in to step 3, which is understanding the chemistry. And this was quite exciting because, you know, it talks a lot about the issue of interdisciplinarity, which is something which is being pushed under Education 5.0. That when you work in a field like, you know, medicinal, when you're working with medicinal plants, one person cannot answer all the questions. And there is that need for that multidisciplinary approach. So it's an opportunity actually through such research to be able to cultivate multidisciplinary approaches to research, especially, you know, concerning these plants. So it got us to work with colleagues from Botswana, organic chemists and other people, and we're able to identify quite a number of alkaloids, one of which had never been identified in this particular plant species before. But also, in addition, I was excited to have isolated and identified a particular compound which had been identified 100 years ago, and no one else had been able to identify it or to isolate it after that. And that's another opportunity when we're talking about these plants, that we can have these plants come up with what are called lead compounds, or substances which can actually be developed further to individual drugs. Remember what I said earlier, that when you're dealing with plants, you've got a plethora of secondary metabolites or chemicals which have got lots of activity. But what you can actually also do is, if there's one compound of interest, that compound can be worked with individually in the hope that you can come up with a chemical in line with the way that, you know, Western medicine moves. And those are, you know, it's opportunities for research, but also opportunities ultimately to come up with something interesting. So this is one of the compounds that we isolated, which has not been reported elsewhere for this particular plant in particular. And then also what we then did is that we all were very interested to find out, to say, okay, so if I get Mzepetu here in Harare, wakafanana here neMzepetu Mashonaland, what is it, Manikaland, is it the same neMzepetu unowanikwa kwaChivi? And through Bonwo Mutsimba, one of the MSc students, and this is another opportunity again in this particular area of heritage-based science with regards to health. The issue of being able to train postgraduate students. There's a lot to be done. I mean, he was coming from agriculture, and we were interested to find out if the alkaloidal content of this plant was going to be different depending on the different geographical and environmental zones of the country. So another interesting opportunity. And interestingly, we found that for Boophane, it seems as though the alkaloidal content remains the same, regardless of whether you take your plant Kwachiri or you take your plant from Amaron. So afterwards, step 4 is then to try to come up with a dosage form, because ultimately you can also take some sugar and you want to sell it abroad. I wouldn't think it's already good, but what you want to do is to come up with a certain dosage form. And this presents itself with some other interesting opportunities, because there are already people in industry who have specialized in doing these dosage forms and manufacturing. So we got into a partnership with one of the companies, Zimbabwe Pharmaceuticals, and we played around with tablets at first, and then now we have come up with a liquid dosage form, which we are now trying to prepare for registration with the national regulator. Then finally here, I've got a step 5. We need to then understand what the regulatory framework is. The moment that you got all these plants, we get very excited, Zumbani no Shanda or whatever else. But you see, if we want to then take these plants outside our borders, there are some certain regulatory requirements that we have to meet. There are some certain market authorizations that we need to be aware of. And if somebody is doing research in this area, it's actually one of those areas where you can actually get lots of exposure with regards to these regulatory things which are required. And then step 6, of course, is to manufacture and get rich. And this is what we are hoping for. The opportunities here are the opportunities for people who are living, whether kumamisha, whether you can do large-scale farming, but also research comes into play. The agronomists come in, the soil scientists come in. We try to see how we can best maximize whatever compounds are of interest and other things like that. And then the last step, I'd say the last but 5, but this is really the last step, the issue of clinical trials. I know at the moment, when you say Mbani no Shanda, many people will ask you, are there any clinical trials which have been done for it? But you see, the way that we speculate we should proceed is that Mbani have been used for years. And instead of first getting clinical data and then coming up with formulation, what we have found is the best way forward is to first come up with a formulation, get it registered, get market authorization, and then as people are actually using it, you can then start to use accumulate data. And this is the type of approach actually which has been used right now for the COVID crisis. The vaccines that we have, most of them do not have proper complete phase 3 clinical data. And it's pretty normal. The reason why there's a crisis, and as time is moving on, the data from phase 3 trials is actually being increased and accumulated. And I feel, for on heritage-based products, that's the direction that we also take. So do these steps I've been talking about and these opportunities, are they really for real? And well, the answer that I can say to you is ask Mr. Mukwati. Mr. Mukwati is this man who is pictured here. He's a local traditional healer. He's a herbalist who approached the then School of Pharmacy with a plant or a product that he had, which he wanted to put out for market authorization. And we worked with him using these sequences, these processes that I've been telling you about. And he became the first person in Zimbabwe to actually have a product getting market authorization from the Medicines Control Authority of Zimbabwe. So there's this product, it's called MCARs or something like that, which is used for the management of genital warts. So I think in those few slides and in 15 minutes or so, that's really what I wanted to bring out, there are lots of opportunities which come with the approach, and this Boophane story was just a way to try and demonstrate what some of the opportunities are. Otherwise, thank you very much for listening. May God bless you all and stay safe. I think I am done.
Thank you, Prof. Tagwire. I think this was, it's always good when I listen to the Boophane story again. And I remember the first time when you talked about it, you gave the example of a person who took the Boophane and suddenly was hallucinating and seeing rivers where there are no rivers. I always remember that one. Yeah, but thanks a lot. I think there are a few points that we understood there. I personally got that there is a very little difference between poison and medicine, and so the right dose determines whether it becomes poison or medicine. And I think you brought out quite well the issue of interdisciplinarity and multidisciplinarity of research. Most of us would really just go at it ourselves. And I think that's a very dangerous approach. It's important, I think, for us to really join hands and work as teams. I think the current notion is for us to do research groups and all that. I think that is very important. Then where I have questions is on the role of MCARs, which I understand very well, that someone needs to be regulating these things. But maybe do we have an alternative to MCARs, or can we improve how they do it? I don't know. And the next one is on the clinical trials that you alluded to, which I said, I always say, if Kanagwenzredu Richishanda, do we still have to go through what we already know that it works by way of clinical trials? So I think that's where we are probably curtailing our innovation using our herbs and heritage, where we are saying you still need to go through clinical trials and so forth. Can we have an alternative to MCARs and to the clinical trials for things like the herbal remedies that we use?
All right. Thank you very much for that question, Professor. So I will start with addressing the issue of MCARs. So drug regulation came into being almost 100 years or so ago. And unfortunately, it's when crises occur that we start to really value or see the place of regulators. There are several stories in the literature. I think people can just Google and look through. But I mean, one of the major ones was when a certain pharmaceutical company substituted, I think they put diethyl glycol instead of propyl glycol in their formulation, which was meant for children. And hundreds of children died. Others became seriously ill by virtue of that. And it was because of this, as well as other disasters, that the drug regulation, regulatory role got its eminence. So talking maybe specifically about these herbs, when a person goes to see a herbalist and the herbalist prescribes Zumbali, it's well and fine, and the laws of Zimbabwe actually allow that under the Traditional Medical Practitioners Act. So I can have, well, not I as in I, but if I were a herbalist, I could have patients that I actually administer whatever type of plant I want. I could even, at the back of my house, have a mini factory where I'm actually making these capsules or solutions. As long as I can give that patient any problem in the front, what happens is that he will come back to me and I will deal with it. Now, the moment that we now want to give any product to hundreds of people, to thousands of people, to millions of people all at the same time, it then presents certain safety concerns. Because the moment that now there is a small problem with that particular herb, how do we know what the people actually took? Which is why then the authority would then insist on quality to say, okay, if you are saying this is Zumbani yako 5 mils, maybe 1 tablet yako contains what quantity of Zumbani. So if there's a disaster now, somebody has taken 2 tablets of Zumbani in Manicaland, we can be able to work backwards together with the clinicians to see how best to help that particular person. So issues of quality and safety become very, very paramount. Whereas if I'm dealing with 1 or 2 patients, I can, work with it, at that particular point. But the moment that I've exposed all these people, then there have to be documents to support what it is that I have and to know if any disasters occur, how best I'm going to work with it. I'm not sure, I hope that came out a bit clear. And then on the issue of clinical trials, like I said during my talk, the way I feel we should, maybe to start with, clinical trials are the ultimate yardstick. You can't say definitively that something works unless you have taken it into a lab. Okay, for example, if we see a ruler or a stick, we can look at it and say, this stick is about 30 centimeters or 14 centimeters. But until you use a ruler, which has been, calibrated, you can't say for certain that it is actually 30 centimeters. You can guess. So ultimately, what clinical trials do, especially the phase 3 randomized control clinical trials, is to let us know that compared to either a placebo or compared to a drug that we actually know, this particular plant has got advantages, or they compare, they are the same. But then the approach which I feel we should move as a country with regards to medicinal medicines, to start off with, Medicines Control Authority does not require for you to have clinical trial data in order for you to get market authorization for complementary medicines. So you don't need any clinical trials. You just have to demonstrate that you can produce your drug or your compound in the same way all the time, and that it is safe. So the approach which I would suggest is that if people think that they've got herbs which work, bring them and let us do what? Let us formulate them. But let us make sure that those herbs are safe. Let us find out what is already known about them. How do these herbs interact with other drugs? Because I wonder, maybe it's on HIV drugs. What if the herb that you're taking has got an effect of, increasing the enzymes, therefore reducing your antiretroviral drug concentration and other things like that? But once we do that, then we get market authorization, and then let us put it out. And if it works indeed, people will come back with testimonies. But if we then want to say definitively, this herb is effective for the treatment of, of illness X, then whilst it is on the market, we can then do my clinical trials. As I just finish off this answer, there's a herb which is called St. John's wort. It was used a lot in Europe and in European folklore for the treatment of depression and other related conditions. And what happened is, first of all, they just put it out as a herb, and others would say Shanda, others would say Aishandi. But even as it was being sold, some other people then came and did a systematic randomized controlled clinical trial. And now it is a drug, or it is a herb which is accepted that if somebody's got mild to moderate depression, you can actually give that particular plant. And I've said a lot. I think I'll just stop here now.
Okay, thank you, Prof. I think it's so difficult when you deal with human as subjects. If it's plants like I do, it's easy. They don't talk, so we can test them anyhow. Prof. Chifamba, do you have anything to say?
Not really. Let's get the young people who are there to ask. Anyone with a question?
Prof, I have a question.
Go ahead.
So in regards to the COVID vaccine, are they also going to implement toxicology? Like, are they going to have like toxicological reports as to how effective or how dangerous the vaccines we're getting is? Thank you.
Prof,
I'm not sure if Prof. Tagwire, you heard that.
I heard it partially. I'm just wondering whether I really want to to get into the vaccine issue at the moment. I think I can just comment as an outsider, perhaps,
That's
to
okay.
say that I think for the vaccines which are currently being marketed, including the ones that we got, phase 1, phase 2 data is there. Phase 3 data is coming up in trickles. I mean, we know that Brazil has been using these vaccines. We know that Turkey started using it in the United Arab Emirates. So I think without wanting to entangle myself into, I'll just stop there. Yeah.
Okay. What a nice way to run away, but that's okay, Prof. Bridget, you have your hand up.
Afternoon, Prof. I was wondering, looking at the plant, it contains alkaloids that are more similar to morphine. Can't we do a research whereby we try and extract the alkaloids and try and see if they can behave the same way as morphine?
Thank you very much. That can't be a Bridget, but okay.
That's bad.
So what we have done, if you noticed in the list of pharmacological tests that we have done, one of them that we have looked at is neuropathic pain, which basically can just be treated by compounds which work via the central nervous system. So yes, it can be done. I mean, some of the alkaloids have been tested before and they are known, but the approach that we like to take is to say, let's not move the final compound too far from the plant, itself. But yes, if you've got certain molecules, you can actually work with them individually. There's one PhD student who's working on them individually to actually see if they retain, the activity or not. But what you find is, many people who've worked with these chemical compounds, the more you extract, it would appear that some of them might actually lose activity because of a number of things. Maybe the ones that you extract are the ones which are easily extractable, so they're the ones which jump out. It could also be because there's some synergistic effects which might actually be there from some other compounds which are there. So the simple answer is yes, you could. It's an alkaloid. There are many alkaloids. You could try it out and see how it would go and compare it with morphine.
Thank you. Any other questions, responses? People are silent.
So, Prof,
I don't know.
we were going to, let's say, map out a way for the, let's say what is this one? The Javanica. What can the university do so that we are relevant to the community in Zimbabwe? What path can we take so that, we become center of information for this plant, which is big deal?
Yes, thank you for that question. I think there are several things that the university can do. I mean, we are the center of knowledge. So to start with, I mean, from the botanist's perspective, just simple, easy, palatable public engagement type of information on how do you identify Zumbani compared to some other plants which are there. There might be also some things that we could talk about from a safety perspective. If someone is to pick up Zumbani from a mine area, is it going to be safe? Maybe there's a possibility that there are some heavy metals which are in it. So just letting the public know such things. But coming into the more scientific perspectives, one of the things that we could, take, I'm biased because I am a toxicologist. One of the things that we would take a serious interest in is to say, all right, Zumbani, people are using it. And whether we like it or not, they are using it and they shall continue to use it. And in using it, they are using it in big, huge quantities. The thing that comes to my mind automatically is, okay, what effect does it have to the unborn child? And that could be a research project in itself, where we say, we just want to investigate using simple animal models, mice, rats, rabbits, to say if this animal is exposed to high doses of Zumbani and it gets pregnant, when the child comes out, are there any deformities which will come out and other things which are related to that? The other issue which could come from a toxicological perspective is, okay, if we take this Zumbani, we're taking it several times, what are the long-term consequences? Because pesapaya when was a tiga, and it goes up into your body, and then your body excretes it, isn't it? Goes up and excretes it. But then now if you drink, and then you drink again, and then you drink again, and then you drink again, there's a possibility that there's an accumulation of some substances which might actually ultimately be toxic to you. Then also what we could do as a university is to assist those people who want to come up with the dosage forms of Zumbani, to come up with syrups, to come up with tablets, which are of an acceptable standard, which can be marketed elsewhere with regards to issues of quality. So I mean, in the short, and then also we could do from a clinical perspective, actual clinical studies to say, does Zumbani really have an effect of preventing COVID-19? Do people who've got mild symptoms of COVID-19, do they get better when they drink the Zumbani? And that would be a defined specific clinical study. But also the whole idea of Kunatira, whereas for us, Kunatira has been within our spheres, what do you call our social, cultural spheres. It's not such a not widely used elsewhere. As a result, then the WHO would be quick to say it doesn't work. But we ought to be writing that narrative. We ought to be saying, does it work or does it not work ourselves, not being dictated by other people. So there's a lot that the university can actually do with regards to this Zumbani and actually continue to be relevant to the community that we are supposed to be serving. Also, I mean, people are taking Zumbani now, but others are taking Zumbani, but they're on antihypertensive drugs. Another quick question that comes to mind is, so what does it do to the other drugs which they are taking? Does Zumbani affect drug metabolism? Does it cause inhibition of enzymes or it causes induction of enzymes? We've heard the community say that women who are on Zumbani and on oral contraceptives, apparently as they seem to be getting pregnant. And the question that comes to mind is, can we actually be able to demonstrate whether this is actually it or not in the lab? Some simple studies which biochemists could actually do and biologists. Otherwise, yeah, I think in a nutshell, that's what I would say. I mean, it's an interesting topic that we can talk for the whole day, but yeah, we'll just leave it at that for now.
Yeah.
Facebook. You want to comment on anything there?
Okay. Oh yes, this was a Facebook post which I did several weeks ago, and basically from this, I was trying to demonstrate and show that if you look at those, this is Zumbani, this is not Zumbani, this is Zumbani, this is not Zumbani, this is not Zumbani. So what is happening now in society is that we've now got quasi-experts. Everybody is an expert. And some of them are picking, this is Lantana Camara. Taking these plants, they smell almost the same to an untrained nose. If you've got this form which has got white flowers like this, if you do not know, you can actually take this Lantana Camara, which has actually been demonstrated to be toxic, the leaves, as well as the green berries, if taken for a significant period of time, can actually lead to renal failure as well as liver damage. So basically through this, I was just trying to make people aware that if you are going to pick Zimbani, make sure you know what you are doing. Because if you make a mistake and pick the other one, you could actually suffer some toxicological effects, you know, in the days or months to come.
Thank you, Prof. Tagure. I think, yeah, people always want to listen to this the whole day. I would. But I think some few pointers that you brought out, I think is there are quite a number of projects that people can embark on and research and get further information. Obviously, the university is really keen to support, particularly if we can widen these herbs. Now people are talking of a whole lot of herbs, Mfantichimuka, you know, and other things. You just see a person, this one is no, this one is no rapper is. I think it's high time we really harness some of these things and have a pool of experts. We are thinking of establishing a herbal institute to try and organize such kind of research. So yes, we do need experts. We do need people who can really push this agenda forward. If we have so much richness in our herbs, it's high time we can also really make use of them, harness them, and make sure that we have things that we can use. I don't see any more hands. Just to tell people that we do have these Zoom meetings every 12 o'clock every day. We try to make sure that they're exciting. So tomorrow we'll meet again at 12 o'clock as we have another Zoom meeting on heritage-based something. This is obviously from the Research and Innovation Directorate who are driving this. Our aim is to try and generate debate, generate researchable areas, generate interest in research and innovation. So we bring in professors, we bring in people with expertise, we bring in students who can really teach us, who can really talk to us about certain things. So make a date with us tomorrow at 12 and every day thereafter we will be there. I think Prof. Chifamba, Prof. Tagure, and other people can have the link. The link is the same every day at 12 o'clock. So I think it leaves me to thank Prof. Tagure for such an insightful Boophane talk again. Prof. Chifamba, you have something to say?
Kaka, I wanted to ask his professor, I don't know where he has disappeared to. His hand was up.
Oh, okay. Okay, has he disappeared or is there?
He wanted to ask his professor.
Oh, Chicago.
Yeah,
Mm.
thank you. No, I didn't have a question. I was just clapping you and say thank you very much for this Zoom meeting. It was a nice.
All right, Chikaka, thank you. And thank you, everyone. We'll meet again at 12 tomorrow. Have a good day and please stay safe and do everything that makes us stay safe. Thank you very much.
Thank you very much, Prof.
Thank you.
Thank you, Prof.
Thank you.